Yes, sir. Now is it visible?
It's okay, you can start.
Okay. Good afternoon, everybody. My name is Saksham. I am doing PhD under the supervision of Professor B.S. Kayat and co-supervision of Dr. Rakesh Kumar from Department of Chemistry, Dr. B.R. Ambedkar National Institute of Technology, Jalandhar, Punjab, India. Today's my topic of presentation is evaluation of physicochemical properties of hybrid bio-based derived adsorbent. Content of today's presentations are introduction, experimental, resultant discussion, conclusion, and acknowledgements. Now, there are many kind of food-based sources. Food-based sources mainly comes from institutional, agricultural-based residential areas such as household, industrial-based like meat, poultry, egg, and seafood processing, cereal, pulses, and beverages industries. Commercials like supermarkets, supercenters, food wholesales, and hotel and restaurant. And institutional waste comes mainly from hospitals, nursing home, military installations, office buildings, and college and universities. These agricultural waste residual burning can lead to serious effect on environment and can cause global warming, emission of greenhouse
Sorry,
gases.
but we can see only one slide, only the first slide. Don't pass the slide. Okay. You can speak.
Now, is it visible?
We can see the fourth slide. Slide 4.
Okay. Residual burning can lead to loss of carbon, loss of soil flora and fauna, deteriorate soil fertility, loss of plant nutrients and biodiversity. Now, these rice straw, wheat straw, and orange peel, potato peels, soybean oil cake, peanut oil cake are the food waste. I mean, these can undergo microbial fermentation and can produce biofuels, biofertilizer, TEMP, animal feed, antibiotics, and many chemicals. So the current research work utilize the eco-friendly waste to wealth approach to address the environment hazards caused by the combustion of wheat straw and disposal of orange peels as bio-waste. Next is the extraction of pectin. Here we utilize the
We can see only the slide 4.
Actually, these slides are not moving.
You have to move the slides.
I'm moving it. Actually, these are moving in my screen, but not. Okay. Next is the extraction. Here we have used orange peels for the extraction.
You have to split the presentation, not only the screen. It is a problem of screen.
Sir, is it visible? I am showing my sixth slide. Extraction of pectin.
We can see the slide 6.
Okay.
Only the slide 6.
Extraction of pectin was done from orange peel, that is a food waste, and for this process we have We have dipped orange peel in water bath for approximately 1 hour in mother liquid of citric acid, and after that the filtration was carried out. And after filtration, precipitation of pectin is carried out with the help of acetone, and after filtration we get jelly pectin, and this jelly pectin is allowed to dry in hot air for approximately 24 hours to get dry pectin. Next is the extraction of nanocellulose. For the extraction of nanocellulose, here we have used wheat straw, that is the agricultural waste, and um, this wheat straw is converted into nanocellulose through three processes. First is the alkali treatment. For alkali treatment, it is dipped inside the NH solution to remove its hemicellulose, and after that, it undergo delignification process mm, under two steps, and we get cellulose, and after that this cellulose undergo acid hydrolysis to convert it into nanocellulose with the help of sulfuric acid. Now, these extracted pectin, biowaste-derived pectin and nanocellulose are mixed together to get a hybrid backbone and soaked overnight, and then itaconic acid, which act as monomer, and MDA, which act as cross-linker, is added to it with continuous stirring, and after that ammonium persulfate is added to the reaction mixture to initiate the reaction, and this whole reaction mixture is allowed to stand in hot air oven for approximately 3 hours to get semi-IPN, and after drying we get semi-IPN. Um, the process of optimization through RSM technique. Now, this semi-IPN is preparation of semi-IPN is optimized through RSM, in which there are 7 independent variables like monomer concentration, cross-linker concentration, reaction initiator, solvent, time, temperature, pH, and these all reaction variables are varied from minimum range to maximum range. And then ANOVA is applied to these reaction parameters, and we get standard deviation of approximately 0.0052, and adjusted R square value is 0.9680, and predicted R square value is 0.9611. From RSM, we get there are only three reaction parameters which are affecting here the effectively affecting the percentage graphic, that is the cross-linker concentration, monomer concentration, and initiator concentration. These graphs are showing 3D graph between the cross-linker concentration, monomer concentration, and 3D graph between the initiator and monomer concentration, and maximum percentage graphic was obtained at approximately 144 percentage. Further, the the samples were morphologically analyzed under the SEM, in which SEM images of pectin showed the smooth surface with occasional irregularities, and meanwhile nanocellulose displays a rough fibrous texture, and the final hybrid material formed by mixing of these two, pectin and nanocellulose, show unique rough texture, and the SEM image of SEM images shows the globular-like structure with significant level of porosity and roughness, and also show the interconnectivity due to the grafting process. Next is the elemental analysis through the EDX, in which pectin, hybrid, and nanocellulose mainly show the percentage of carbon and oxygen, as these are polysaccharide. And while in case of semi-IPN, due to the cross-linking of MBA, nitrogen is introduced at weight percentage of 12.43% and atomic percentage of 11.45%, which shows the cross-linking of MBA in the graft copolymer. Next is the FTIR analysis of the samples, in which nanocellulose shows peak at 3340 centimeter inverse, that is due to the OS stretch, and this peak at 2985 and 2894 centimeter inverse is due to CH and CH3 stretch in the nanocellulose. The peak at 1030 centimeter inverse and 895. These are the characteristic peak of the nanocellulose, in which 895 centimeter inverse peak corresponds to the beta glycosidic linkage of the cellulose, and the 1030 centimeter inverse peak corresponds to the COC bond of the O-glycosidic linkage in the nanocellulose. While in case of pectin, the peak at 1739 and 1052 are the characteristic peaks in the pectin. And this peak at 1739 centimeter inverse is due to the CO stretching of glutamic acid in the pectin, and this peak at 1053 centimeter inverse is due to the COCH3 bending in the pectin. In case of hybrid, these show all the characteristic peaks of pectin and nanocellulose, while
We have lost the slide. We have totally lost the slides. But we have lost you. The image is fixed. I think there is a problem. Yeah. It's a connection problem. Son image est fixe.